Hi, everyone, and welcome to episode 636 of the People Behind the Science podcast. Today, we are thrilled to be rebroadcasting our interview with Dr. Russell Foster, and this interview was originally published on March 7th of 2016. Listeners, Russell is a distinguished neuroscientist who has focused his research on understanding how light is detected and processed by photoreceptors to regulate circadian rhythms, as well as how circadian rhythms are generated and what happens when these systems break down with disease or abnormal environmental conditions. His research group is credited with discovering a new class of photoreceptors in the vertebrate eye that regulate circadian rhythms, sleep, heart rate, and pupil constriction. And in our interview, listeners, Russell talks more about his life and research. So get ready to get inspired in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Russell Foster. So, Russell, welcome to our show. How are you?
I'm very well, and thank you very much, Marie.
Well, we are delighted to have you with us today, and I want to start by giving our listeners just a little bit of background information about you. So, listeners, Russell is professor and chair of Circadian Neuroscience, Supernumerary Fellow in Circadian Neuroscience, head of the Department of Ophthalmology, a Nicholas Kurti Senior Fellow, head of the Nuffield Laboratory of Ophthalmology, and head of the Sleep and Circadian Neurosciences Institute at Brasenose College at the University of Oxford. He received his PhD in neuroscience from the University of Bristol, and Russell worked at the National Science Foundation Center for Biological Timing at the University of Virginia, and afterwards served on the faculty at Imperial College before accepting a position at the University of Oxford, where he has now been for 10 years. And Russell is a Fellow of the Royal Society, and in 2015, he received the honor of an appointment of Commander of the Order of the British Empire. He has also received many other national and international awards for his accomplishments in science, including Japan's Honma Prize, the USA's Cogan Award, Harvard's Farrell Prize, as well as the UK's Zoological Society Scientific Medal and Edridge-Green Medal from the Royal College of Ophthalmologists. In addition, Russell is the author of a couple of different popular science books, including Rhythms of Life, Seasons of Life, and most recently, Sleep, a very short introduction. And listeners, Russell is here with us today to talk about his journey through life and science. So Russell, today we want to get to know you, both as a scientist, but also as a person. So can you start by telling us what you like to do when you're not very busy doing science?
Well, it's great to get together with the family, of course. My children are in their 20s. In fact, my eldest daughter, she's currently in Chicago. She's coming home fairly shortly. But it's lovely to get the family together and around the dining room table and talk and laughter. That's fantastic. If it's not with family, then I'm passionate about music. I love opera, Mozart. I love Wagner as well, but also the symphonies. And outdoors, I love the sea. And so we have a small cottage down in Lyme Regis, which is this incredible place where Mary Anning found all those Jurassic fossils. And so we get down to Dorset as much as we can, and I have a sea kayak, and so go out on the sea. I love to be out on the sea, and I'm a keen swimmer, so water is always very attractive to me.
Well, it sounds like you've got a little bit of everything there. You've got some family, some music, some ocean. Great to get a window into your life outside of science. And now, Russell, I want to talk a little bit about your science next. So can you give us the high-level look at what you do and how you describe what you do to someone who might not be familiar with science in your field?
My broad interests in how body clocks, circadian rhythms, and the sleep-wake cycle are both generated within the central nervous system and how they're regulated, and particularly how they're regulated by light. And so trying to understand the fundamental neuroscience of what makes us tick, but also to try and understand mechanistically what happens when these systems fall apart. So many health conditions, ranging from mental illness to eye disease, can hugely disrupt the sleep-wake timing systems. And I want to understand what precisely is going wrong when sleep is disrupted. Then to use that mechanistic understanding of disruption to think about evidence-based interventions and therapies to try and stabilize sleep-wake. Disrupted sleep is so much more than the inconvenience of not sleeping at a desired time, but is associated with a whole range of problems. The ones we've experienced, all of us, which is the cognitive impairment, the failure to process information when we're tired, or impulsivity, or a whole range of loss of sense of humor are all characteristics of being tired and sleep disrupted. But chronic sleep disruption is associated with immune suppression, raised infection, raised cardiovascular disease, increased stress, anxiety, and in fact, sleep disruption most recently has been associated with sort of nudging you if you're predisposed into a psychiatric illness state. So, in brief, understanding the mechanisms whereby sleeping clocks are generated, what happens when they go wrong, and using that information to try and understand ways in which we can correct them to improve overall quality of life for not only the individual, but of course the people that they live with.
Well, Russell, you've given us some very convincing reasons of why we should be sleeping more. But I know you have a fantastic TED Talk out there as well, where you talk about why do we sleep. And I'm a quote person myself. You pulled out some great sleep quotes in that TED Talk. But I want to hear a motivational quote from you today. So can you tell us what is one of your favorite motivational quotes that really keeps you going through all of the busyness of a day?
Oh, I would definitely say keep on kicking the door until the buggers let you in. Don't be put off. Certainly with some research that we did early on in my career, where we discovered that there was a completely new photoreceptor system, a new light sensor within the eye. And when we first proposed this idea, sort of people were pretty hostile, saying, look, we've been studying the eye for 150 years. Are you seriously telling us that we've missed an entire class of photoreceptor? And of course, I was fairly young and I suppose a bit more bullish and sort of said yes. And we did, of course, better and better experiments to actually prove it. But it was tricky. I mean, grants were not funded, papers were not published, because the dogma was that this could not be right. And so what I learned fairly early on from, in fact, some wonderful colleagues when I was based at the University of Virginia, notably Mike Menaker, which is, you're right, have the faith in your science and just carry on. If the science is solid and you know you've done the right experiments, then don't be ashamed to carry on. And I was lucky enough to have that opportunity.
Absolutely. I think when you're on the edge of a field, when you're making these exciting new discoveries, it can be very hard to get the rest of the field to accept them sometimes. So it's great to have these colleagues, these people behind you to support you along the way. And I love talking about the people who have supported scientists along the way. So can you share with us who are some of these people who you've looked up to or who've really made an impact on your career that you consider role models?
Michael Menaker was my chairman, in fact, when I was based at the University of Virginia. And we fired off a grant to one particular institution, and it came back, and it was just shredded and absolutely unfundable. I was called to Mike's office, and I thought, oh, my God, I'm going to be given a real dressing down. Right. And he said, look, you're right. The data are beautiful. Just submit it to another granting body. I said, what, just turn it around? He said, yes, just turn it around. And it was the most extraordinary thing because I got a call from this other granting body saying, Dr. Foster, we'd just like to say that your application has come top of the applications. Oh, wow. And we wondered if you would mind binding it up and taking it to the Society for Neuroscience meeting as an example of how research grant should be written. Now, this was the same grant.
That is phenomenal.
That had been completely shredded. That was, I think, an incredibly important lesson. First of all, you do keep on kicking the door until they let you in, but also to have had that support by a very distinguished and very senior member of the faculty was also immensely empowering. I was privileged to have had Mike as a colleague. And of course, we're still good friends. I just saw him fairly recently in Copenhagen at a meeting.
Fantastic. Well, I think sometimes, too, you just have to find the right audience, whether it's a funding mechanism, a paper, a presentation. Sometimes it'll go well in one place and horribly in another. So,
well, that's very true. Yes,
definitely. Well, wonderful to hear about one of these people who's had a big impact on you and helped you along your journey. But I want to hear about how your journey began. Can you tell us why science? And what was your earliest memory of kind of falling in love with science?
My earliest memory is a very early one, and it must be before I was 4 or 5. Because I remember, and I can see it in my mind now, of lying on the ground, looking at a lizard basking on a rock. And I just spent a long time just looking. And I was fascinated, like all kids, of course, by dinosaurs, the How and Why Wonder Book of Dinosaurs, which is a book I've still got. And I collected insects. I loved to swim, so we would swim in the sea and get our fishing nets out. And it was just that connection with biology. So in a sense, I always knew I wanted to do something with biology. Then I thought, well, I'm probably going to do marine biology because I swam. If you're interested in biology and you swam, then the obvious thing is to do marine biology.
I was in the same boat myself, yeah.
And then I went to university and I studied zoology. And as I say, I thought I'd do marine biology. And then we did some physiology labs, and we recorded the light responses from the eye of a locust. And I just thought it was so amazingly cool that you could actually study the physiology of a sensory system. That was part of the switch. Then another switch was reading an extraordinary book called J.Z. Jung's Life of the Vertebrates. In that book, he sort of goes through the vertebrates and talks about their overall biology and their structure. And there's a chapter all about lampreys. And lampreys not only have lateral eyes for vision, but they also have this third eye, their parietal eye, which they use for a whole variety of different detection tasks. So, for example, as a shadow response, they will move in response to a shadow. And I just thought it was so extraordinary that there was this weird photoreceptor system that I didn't know anything about. I went into the lab and spoke to Alan Roberts, who's another inspiration for me, and I did a project with Alan on recording from the third eye, the parietal eye, of amphibian embryos and studying the light responses of those extraordinary pineal eyes. And so I got into photoreceptors and weird photoreceptors as an undergraduate. And in those days, I was lucky to have had people who were immensely supportive and who nurtured your enthusiasm. And that was just fantastic.
Certainly. And I think some of those early laboratory experiences, kind of taking science out of the classroom, are so big in terms of career changers for people, kind of helping them decide what they want to do and showing them how exciting science can be.
And it was proper research. We published the paper. That was my first paper. And it was so immensely exciting that as an undergraduate, I had a mentor and we did some great science and we published it. And it was peer-reviewed and it still stands there. It's my first bit of science.
That is fantastic. Well, wonderful to hear about how this whole journey began for you, Russell. But now, can you tell us, what do you have going on in the laboratory right now that you are just the most excited about?
Gosh, where to start? There's so many amazing things going on. One of the things that we've studied, of course, has been these new photoreceptors in the eye, these photosensitive retinal ganglion cells. And these are the receptors that detect brightness at dawn and dusk and then set the body clock to local time. The classic mismatch between internal and external time will be jet lag. Essentially, you get over jet lag as a result of exposure to the new light-dark cycle. Now, we've moved on from understanding how these photoreceptors work, how they detect light, and that's remarkable in of itself, but also how the light information from those receptors interacts with the molecular biological clock. So what is light information actually doing to the clock? We've discovered a whole bunch of key elements which are really important in transducing that light information and turning it into a signal that then will actually shift the clock. It will actually change the pattern of clock gene expression and align the molecular clock to the external world. And that's been really very exciting. One of the questions has been, well, why does it take so long to get over jet lag? You move across multiple time zones. Why do you take about a day for every time zone you've crossed? There must be effectively a break of the effects of light on the clock. And to cut a long story short, we discovered a particular protein, which when you remove it, or when you knock this protein down and you shift the light-dark cycle, the mouse will move to the new light-dark cycle immediately.
Really?
So it doesn't get jet lag. And so that's, I think, amazingly exciting because, first of all, it's told us something fundamental about the nature of how light interacts with the molecular clockwork, but it also potentially provides us with an interesting target. What we've effectively done is increase the sensitivity of the clock to light. Now, that could be important for a large number of different groups. So in the elderly, for example, who are, let's say, in a nursing home, not exposed to particularly strong light, and indeed it seems for some reason which we don't understand, clock systems get less sensitive as we age. Maybe a drug could actually increase the light sensitivity of the clock, which means it would be much easier to keep the sleep-wake cycle of the elderly on track and therefore mitigate some of the sleep disruption and all of the problems that you get from sleep disruption. That's one of the projects, which is understanding, again, the fundamental mechanisms, but then applying that to the broader clinical realm. Another area, of course, would be the impact of eye disease on human sleep-wake timing. And so working with my colleagues in the eye hospital here in Oxford and other centres around the UK and indeed the world, we're interested in how different sorts of eye disease disrupt the sleep-wake cycle. And we're developing evidence-based guidelines to say, well, if you have this level, let's say, of glaucoma and this level of eye damage, then this is the likely consequence, and you need to take all of these things into consideration. I suppose the bottom line is, because we discovered you can lose your visual sense, you can lose your rods and cones, but these other receptors, these photosensitive retinal ganglion cells, can be working imperfectly fine. So you can lose your sense of vision, but not your sense of time. Many ophthalmologists don't quite get that yet. They'll say, you're visually blind, well, that's it, there's nothing more I can do for you, I'm sorry, without sort of saying, well, actually, those photosensitive ganglion cells are still working. What you need to do is, even though you can't necessarily sense that light visually, get out, try and expose yourself to sufficient daytime light to make sure the body clock is locked onto the external world. If you don't get that daily signal, then, of course, the individual will drift through time, in a sense, experience unremitting jet lag for the rest of their lives. So those are some of the photoreceptor stories emerging. And one of the other major projects is trying to understand the nature of sleep-wake disruption in mental illness. In conditions like schizophrenia, we've shown that the sleep-wake pattern is completely smashed. It is extraordinarily disrupted. Sleep disruption in schizophrenia has been known since the 1880s, is when it was described fully, but it was largely dismissed from the 1970s onwards as, well, it's an artifact of the antipsychotics, or these individuals don't have a job, so no wonder they get up late, miss my clinic, don't have friends. It was completely disrupted. And we came up with a sort of a new way of thinking about this, which is that because the sleep systems draw from multiple neurotransmitter systems in the brain and multiple brain structures, it may well be that normal mental health and normal sleep share overlapping neural networks within the brain. And we were able to test that hypothesis by taking a gene that was originally linked to human schizophrenia and looking at a mutated form of that gene in a mouse and asking what's happened to the sleep-wake of that mouse. And in fact, the sleep-wake is massively disrupted. So shown empirically that there is a genuine mechanistic overlap between how the brain generates good sleep and how the brain generates good mental health. And of course, the sleep disruption may exacerbate the mental health condition, and the mental health condition may exacerbate the sleep disruption. But the origins of the problem lie in those overlapping pathways. And that mechanistic insight, I think, is leading us to think of new ways of thinking about the treatment of sleep disruption in mental illness. And that's also, I think, turning out to be really quite exciting.
Well, this is fascinating work. Both of these lines of research talking about the photoreceptor work, also sort of the implications for mental illness of sleep. And I'm curious, as a scientist who studies sleep and circadian rhythm, can you tell us a little bit about your own sleep routine and sleep patterns? You probably get this question all the time.
Well, as a student, I was very late. I'm still delayed. So I used to go to bed late and get up very late. And sometimes I would work until dawn. Now, as we age, we tend to get earlier in time. We go from owls to becoming more like larks. And now I still probably go to bed between midnight and 1 o'clock in the morning. And I think one of the great advantages of being a professor and having some meager power is you do decide what time the meetings take place. And in my lab, at least, meetings don't take place until 10 o'clock in the morning. And of course, as my meetings are generally with bright young people who have delayed body clocks anyway, a 10 o'clock meeting is perfectly civilized. So I'm lucky enough to be able to map my body clock type onto the demands of work. I mean, I won't leave the lab often until 8 or 9 or perhaps even 10 o'clock in the evening, and that's just because I love to do it. I have the luxury of being able to do that. I suppose the tragedy is that many people don't, and they are forced to work against their body clock type, and with all of the problems. I mean, night shift work is a classic example. The life of night shift workers is really grim, and that can be up to, 18% in the developed world, and I understand now that in China, about 28% of the population are doing sustained night shift work. And the health consequences are really very serious, and we're doing nothing for that group of workers. And there's stuff that we could do now. We're not going to put that 24/7 genie back in its bottle, but knowing that our vigilance will be impaired at night, then we can use electronic devices to warn you that your vigilance is impaired, not only in the workplace, but also on the drive from work back home, so you're less likely to have an accident. Knowing that the gut can't process food properly at night, we can provide food that is easier to digest. Knowing that night shift workers are prone to obesity and diabetes too, we can stop filling them up with fast food, but also give them a nutrition that is more appropriate for the gut at that particular time of day. So there are things that we can do to mitigate some of the effects of night shift work. Now, of course, we drifted from my sleeping habits to night shift work. I slightly got off track there, but I hope it's made the point that we all have very different sleep-wake patterns, and that's another very interesting question.
Absolutely.
If you look at a population of mice or anything else in the wild, the sleep-wake pattern is pretty narrow. But as a species, we have huge diversity. There are morning types, there are extreme late types, and of course our sleep-wake patterns change as we age. And I think that's a very interesting biological question. We don't really understand why that's the case.
Certainly. And I think just in daily life, a lot of us take this sleep wake pattern for granted. So it's interesting to talk about these ramifications, some of the problems that people might face, for example, if they are a night shift worker. So thank you for sharing those with us, Russell. And I know earlier we mentioned this photosensitive cell that you discovered, this entirely new cell, as an example of your success. But I love talking about all of the successes in science. So I want you to share another success story with us, one of these really great moments in your career.
I think the opportunity of working with bright young scientists and getting their careers started. I think there's nothing more exciting or fulfilling that you've done your stuff, you've created an environment in which others can thrive. And that, to me, is immensely exciting. And part of the reason I wanted to develop this new Sleep and Circadian Neuroscience Institute is to provide the infrastructure for these bright young minds to thrive. Because seeing how their careers develop and seeing the discoveries that they make, I think is immensely rewarding, for me at least.
Certainly. So can you tell us about how many people have you been able to train in your career?
I suppose numbers of PhD students, it must be coming up to 15, maybe even 20 PhD students, and large numbers of postdocs. In a sense, it's strange because I tend to think of them as colleagues rather than my PhD student or my postdoc. So that's why I'm hesitating on the numbers, because these are not notches on the belt.
Right.
These are people I've just been absolutely honored and delighted to work with. And one of the first postdoc was a chap called Rob Lucas, who's now full professor at the University of Manchester and doing wonderful, amazing things there. We've got a brilliant young scientist, Arti Yaganath, who has published a recent Cell paper and is doing some amazing work. And you see that they're applying for fellowships and they're going off and doing things. And there's lots of individuals that one could mention. And they've been fantastic colleagues.
Well, I love that you have really kind of honed in on these people becoming your colleagues, your peers. And I think this is one of the great parts of the scientific process is you work with these people, perhaps when you're training them, but then they do become your colleagues. You see them at conferences, you keep in touch, and it's just kind of a wonderful community that you form.
Yes. I think, again, we're not only do we get to decide what time we go to bed and get up to some extent, but I think we're immensely privileged because we are working with some just extraordinarily bright, enthusiastic minds.
Certainly. Now, we've talked about a lot of the great things about careers in science, in your career specifically, but I want to talk about some of the dark times. I know every scientist has them, these moments where things go wrong, you fail, you are so frustrated with something. And it's important to kind of get through these tough spots because they happen a lot in science, but it's also important to share them with the world so people understand all of the things that a science career encompasses. So can you take us to one of your own dark days, Russell, and share what happened and how you got through it?
It's quite interesting. We talked about my interest in music, and when Rossini was asked about the music of Richard Wagner, he responded, there were some wonderful moments, but appalling half hours. And science can be very much like that. There are these wonderful moments. You've got that discovery, and then you say, right, I'm going to write it up and turn it into a paper and then a grant proposal. And I remember when I moved back from the States to the UK, I'd sort of cracked it in the States. People understood what I was about, what we were trying to do. And the grant income had followed after that rocky start. And then I thought, right, well, I invited back and joined Imperial College and fired off some grant proposals, which were very similar to the ones I already had funded in the States, but I couldn't bring those with me. And they were rejected. They were just turned down. And I thought, oh, my goodness, I'm going to have to go the whole thing again. Mm-hmm. So I remember getting yet another rejection of a really beautiful grant proposal. And I thought, what the hell am I going to do? And I remember walking around Hyde Park, in fact, in London, this wonderful, great big park, and getting myself in order. And I'd forgotten it was my birthday.
Oh, no.
I got home late, and my wife had organized a wonderful birthday party, which was a complete surprise. And I was feeling wretched. This grant proposal had just crashed. I walked through the door, and they said, happy birthday, and the kids are there, and the family were there, and there was this wonderful, and you think, well, actually, it put it all in perspective. And it goes right back to where we started. The family, essentially, although how frustrating and immensely frustrating it was to have had those grants turned down, to have that support and love, really, of one's family was supersedes and trumps everything. So whilst science can be immensely frustrating, and it is a critical part of one's life, but it is only one part of your life. And the most important thing is, I think, one's friends and family. They've always been immensely important in getting over those frustrations. I have to say, though, my poor long-suffering wife, Lizzie, has seen both the best and the worst of me.
Of course. Well, I think you have a great strategy here, kind of taking a step back and putting it all in perspective. And I think you're absolutely right. Your spouse, your friends, your family, these people are critical sometimes to keeping you sane as a scientist. So.
And grounded. because with some of the success that's evolved, and I feel immensely privileged in that regard, but having somebody who's actually can put things also in perspective, so yeah, that's fine, but now you've got to cook the tea and do the sort of the normal routine domestic stuff that you do. And I think it all puts it into a proper framework.
Absolutely. Well, wonderful to hear about one of these challenges and your strategy to get out of these tough times. And I know we mentioned early in our introduction that you are an author yourself, in addition to being a scientist. And I love talking about books with everyone we have on our program. So, Russell, can you give us an idea of the type of books you like to read and maybe give us an example of one that you would really recommend we check out?
I did an event just recently, and I was very lucky to do this, with the Royal Society of Literature and the Royal Society on the nature of sleep. And I tried to persuade them to get an author called Jonathan Coe, because he wrote, it must be a decade ago, a book called The House of Sleep. And it is the most wonderful novel about sleep and sleep scientists. And he uses sleep as a metaphor for a whole range of sorts of things. Jonathan Coe spoke, and I then spoke about the science of sleep. But I'm an enormous fan of Jonathan, and I got to meet him. We hung out, and it's such a relief because you never know quite what your heroes are going to be like.
Right.
And I'm happy to say it was one of the highlights of this year. And he's just written another novel, which has just been released in the UK, called Number 11. I just started that, and that's fantastic. So I enjoy literature very much. In fact, it's very interesting. I have my wife to thank for that, because when we first met, she did an English literature degree, and we met, and we exchanged books. And she sent me Nabokov's Pnin, because I admitted that I'd never read a novel. I read biography and history. I didn't quite get novels. And then I read Pnin, and I thought, my God, this is amazing. And I was completely turned on to novels.
Right. What have I been missing?
Absolutely. And I love music and all those things. And this whole new dimension then opened up. And I should say, parenthetically, the book I sent to her was Biology Made Simple, which was perhaps a little offensive. Particularly as she actually got a better grade at when she was 16 in her biology exam than I did.
Oh, wow. Could have been worse. Could have been Sleep for Dummies or something. That's not so bad.
So Anita Bruckner novels, very evocative, and I don't think she's sort of gone out of fashion now. Sebastian Faulks, I think, is an extraordinary novelist. So the broad spectrum, and I must also admit that I've come to Shakespeare fairly late in life, and I now am just completely hooked on Shakespeare. I get it. And it is so extraordinarily rewarding. But I remember we just saw Othello fairly recently. And at the end, Othello is stabbed by a sword. Afterwards, I kept on saying, but what happened? Did he die? Did he get septicemia? I don't know what happened. But just the language. And I'd read some of these things at school, but never really tuned in. And now I'm reading them again, and I'm seeing the plays. And you just see what another set of doors Shakespeare has opened for me. So I love literature. And in fact, one of the great passions is to go away and find a new book. And that's what we try and do for our 2-week summer holidays.
Wonderful. Excellent recommendations, Russell. I'm going to put some of these out on our website for our listeners to find. Things like Number 11, House of Sleep. Great to talk about books with you. And you mentioned that your books are sort of a way for you to escape. You take them with you when you go out on holiday. And I know travel is wonderful. I love being able to escape sort of the average every day for a little while. And I think this is yet another perk of careers in science is you get this opportunity to travel. And especially recently, you've been traveling quite a bit. So can you think back now on all the places that you've been for science and tell us about one of your favorite experiences and where you were?
It has been extraordinary. I mean, last week was Stockholm. The week before that was Boston. The week before that was Abu Dhabi. The week before that was Copenhagen. The week before that it was Nice. In fact, the collective noun for a professor is now an absence.
Right. You live out of your suitcase. Yeah.
Exactly. And my lab look at me very sheepishly. And I've said, look, I'm here until the end of Christmas and I've cleared the first 3 months of next year so we can properly catch up. So, I mean, I love Boston, and I was so honored to get the Farrell Prize recently from Harvard, and that was fantastic. I also got the Feldberg Prize earlier in the year, which allowed me to give 3 different lectures in Germany. I started in Berlin and spoke to our good old friend Peter Hagemann in his institution. And Peter Hagemann has also discovered how algae have these wonderful photoreceptor systems, these Chlamydomonas opsins. And in fact, these photopigments are now being used in optogenetics. And so started off in Berlin, which is a city I absolutely love, and then went down to give some lectures in Freiburg with Dieter Reimann, then finished off in Munich with my old friend Till Röneberg. And it was absolutely hilarious because this is the big lecture at the end of the series. Mm-hmm. And Munich's another great city. But Till, being the person he is, I gave this lecture in one of the ex-royal palaces.
Oh, wow.
Absolutely extraordinary. And the would-be king of Bavaria actually turned up because it's his palace still. And it was this extraordinary dinner and discussion afterwards of science and art and culture and history. This is, I think, the great-great-grandson of Ludwig II. So the trip through Germany was immensely exciting. But also, I was very lucky to go to Abu Dhabi and visit the United Arab Emirates. And I came away being completely enthralled. I met many women scientists who were empowered, who were comfortable with both their gender and their religion. They were very much involved in the organization and running of things, and I was really excited to see that I guess I had a slightly different view of the place before I went, and then came away being much more excited and interested in Abu Dhabi and the United Arab Emirates and seeing the enthusiasm and the energy that they're putting into science in that country.
Certainly. I think some of my favorite travel experiences when I look back are times when I've been surprised, when things haven't quite been as I expected.
Yeah.
Wonderful to hear about your experiences in travel. This tour through Germany sounds amazing.
It was terrific.
What was your favorite thing you saw?
Well, I suppose if I hadn't have done the biological sciences, I think I'd have done history. And I love the history from the Battle of Waterloo through to the 1950s, sort of the modern period. And when you go around Berlin, and so many of the buildings have the pock marks from the bullet holes and the shatter marks from this city that was completely smashed. And all the renovation that's going on and all the modernity is juxtapositioned with this very visible history. And I do hope they don't over-renovate the city, because you get that sense of history. You turn a corner and there's a brick wall, which is completely pockmarked with bullet holes. And talking of books, incidentally, another wonderful book I've just recently read called Leaving Berlin. It's very atmospheric of the city in 1948, or how it must have been. So I think that was something that left a lasting impression. And of course, the Berliners, they just see this every day. But when you are faced with the history of that city in very tangible terms, I think it's quite sobering.
Certainly. I know I myself am really just in awe of the history when you go through anywhere in Europe. Coming from the United States myself, nothing's nearly as old as it is when you go to Europe.
No, but there's still a glorious sense of history and tradition. And certainly spending 8-9 years at the University of Virginia and being in Charlottesville and seeing what Thomas Jefferson achieved and the inspiration and the leadership and the founding, well, of course, one of the founding fathers. So I think America has a younger history, but just as distinguished.
Well, thank you, Russell. Well, it's been great to chat about travel with you. And I know we've talked a little bit earlier in our interview about some of these people who had a big impact on you in science and how you really treasure being able to work with people, some of these bright minds you mentioned in your field. And I think the people are a huge part of what makes science great. And I know there are stereotypes out there about what scientists are like, and we try and break those on our program by sharing the human side of science and just some of the fun and funny things that scientists do when they're together. So can you tell us about maybe a quirky tradition that you've experienced in a laboratory, or just one of these fun or funny memories of your own?
Okay, just a very, very recent one. I mean, you can get a sense of how my colleagues think of me. I just got a brand new, wonderful, fast computer. It's something I was quite proud of. And anyway, some members of the lab came in and they looked at me and they looked at this fantastic, wonderful new computer. And they rather rudely said, "Yes, that's fine, but it's a bit like giving your grandmother a Porsche," which I thought was a little rude, both to grandmothers and me. But that's a good illustration of how they think of me. And of course, and within 5 minutes, I was saying, "Well, can you help me? Sort of, where's the unlock?"
Sheepishly Googling things. Yeah.
Yes, exactly.
Well, that is fantastic. So you mentioned that you have a lull in your travel schedule in the next couple of months. So how do you plan to spend it with your time kind of regrouping, getting back together?
Well, we've got to think about the next stage for this institute. We had a 5-year strategic award from the Wellcome Trust. And although that's got 2 years to run, we need to start thinking about what we're going to do that's new and special next time round. So I want some time to think and interact with my colleagues across the clinical and the basic science spectrum to see how we might position ourselves for a grant application in about 6 months to 12 months' time. I promised to complete with a co-author a 4th book on circadian rhythms, and I said I would do it over the summer. And we got 5 of the 9 chapters done. We've got 4 chapters to complete. I want to work also on getting a bunch of papers written. So it's really regrouping it, catching up, and actually doing the sorts of things that I promised to do. One thing about writing books, and I've always had co-authors, and I think it's very important for me, because if you have a co-author, you can't let them down. It's so very easy not to write a book if you're the only author. There's always a more pressing reason for not spending a week writing a chapter on a book or something.
Sure.
But if you don't do that and you have a co-author, you're letting them down. So I suppose my sort of popular science publications have been driven forward by sheer naked terror of upsetting my very good friends who are co-authors. And it's critical that you highly respect and value your co-authors, because then it's the guilt. It drives you forward.
Guilt can be very powerful, absolutely.
Yes, indeed.
Well, now we are all your accountability buddies for this fourth book. So, well, wonderful to hear about some of the plans that you have coming up. And I know we've talked about some of the research you've done, some of these things that you hope to do in the next couple months. But I want to know about the big question, Russell. If we gave you everything, if things like funding and staff and technology and feasibility were not a problem, what is the question that you would want to answer?
I think it's got to be sleep and why we sleep and what's going on in the brain whilst we sleep. Our understanding of sleep has been transformed in recent years, and one of my bright young colleagues, Vlad Lyazovsky, has discovered that sleep is not a global event, so that we tend to think of either being in the sleep state or the non-sleep state. But he's found that parts of the brain, while you're functioning, while you're awake, can go to sleep. They actually go into the sleep state. So this is just one illustration for why does that happen. How do we use sleep to not only consolidate memory, but also use information? How is the brain capable of taking information and solving problems, coming up with innovative solutions to complex problems? Because that's one of the great things that's going on while we sleep. So how the brain actually does that, and in a sense makes us the most extraordinary species that we are. And sleep is such a fundamental point of that. I mean, it's worth bearing in mind that when we celebrate our 60th wedding anniversary, 21 and a half of those 60 years, you'd have probably been asleep. So I'm not sure if you can take full credit for that 60 years if 21 and a half years have been fully asleep. However, the point is, the quality of those 21 and a half years of sleep will dictate to a large extent the quality of the waking experience. I think it would be a major initiative in understanding what's going on in the brain whilst we sleep, and indeed how that's come about, the evolution and the biology of sleep. That's what really fascinates me at the moment.
Well, Russell, you have given us a lot to think about, and I know I'm going to have trouble quieting my mind next time I snuggle up to go to sleep. So thank you for that.
It's been a great privilege. Thank you.
Absolutely. Now, we'd like to give our listeners a little piece of advice, something from our guest that they can apply to their lives, think about as they're going through their careers. So, Russell, can you share with us one parting piece of advice, or maybe just an inspiring note that you'd like to end our conversation on?
Oh, that would be keep a sense of proportion and always, always be prepared to laugh at yourself. Never take yourself too seriously. It's all about maintaining a sense of humor. And encourage your friends to laugh at you.
I like it, Russell. Definitely have to keep that sense of humor very important in science. Now, can you tell our listeners where they can go to learn more about you and your work?
Yes, we have the Sleep and Circadian Neuroscience Institute. So if you do just a search on sleep and circadian Oxford, then the website pops up. One we've talked about, Sleep, A Very Short Introduction. That's a new book and covers much of the sorts of areas that we sort of talked about. And of course, I'm very happy to answer questions via email. And you can just find me by Googling the Oxford website. And I might not answer immediately, but I'll do my very best.
Perfect. Well, Russell, thanks again for your time today. It's been a pleasure.
Thank you.
And listeners, thank you so much for joining us. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.